So basically, I would like to give you just some kind of summary or preview of the research, cutting-edge research, which is going on in the world, I guess, in terms of genomics. So as you know that genomics had changed everything. Genomics means sequencing the human genome or sequencing the genomes of people who have rare diseases or any other diseases. And this technology has been evolving at a very rapid pace. The first human genome was sequenced in 2002, and the cost was $3 billion. Today, anybody around here who has $1,500 in their pocket can sequence their genome. And the first genome took like 12 years to sequence. Currently, we can sequence the whole genome at a cost of $1,500 in just 3 days. So this is the technological advancement we are talking about. And the first genome was sequenced with the hope that we will be understanding the genes which are responsible for human diseases. That was the sole purpose, because the genes have all the secrets about us, inheritance as well as how our body functions. So the whole idea was that if we sequence the whole genome, we'll identify these genes and we'll have the codes for life. However, what happened when the first genome was sequenced in 2002, nothing happened. People didn't get any other breakthroughs for a few years until like 2005, until my group actually published the first paper where we identified the gene for age-related macular degeneration. And I think that was the beginning of genomics era, whereas our precision medicine era. So what I'm going to talk about is, I think in 2009 or 2008, when the next-gen sequencing technology came around, the cost of the whole genome sequencing was exorbitant, like $1 million. Nobody had that kind of money. So we decided most of the genes which cause human diseases are actually in the coding region, or only the 1% of the region of the genome which makes proteins. So we thought, what's the point in sequencing 99% when we are going to get 85 to 90% of the gene only in 1% of the genome? So we developed the technology by which we can isolate this particular region which makes proteins, and we started sequencing that, and that's called exome sequencing. This slide just shows that exome procedure. So since then, we have been that technology has been so wonderful in terms of finding disease-causing genes that we have published more than 400 papers, and many of those papers are in Nature Science, which means they are very high-profile publications. So I think most of these genetic disorders, they are rare, because mostly they are in like 50% of the cases, they are in children. Most of the time, these babies are not even born because they are so defective. And even if they are born, they are very abnormal kids. Many of them, they don't reach to the puberty. So that's why finding the patients or populations with rare disease is very rare. So in most of the cases, these are point mutations. That point mutations means there are 3 billion bases in a genome. Out of that, one base really changes. And that base changes the protein function, and that causes the disease. It's a really simple fact if you really think in that way. But to identify that one base which is causing the change, or which has actually changed, is extremely difficult. And I think the technology is improving. It's very, very easy now. So basically, I think we have sequenced almost like 50,000 samples so far. And it's very expensive, it still costs like $300 to sequence one sample. Then we have to do the analysis, then we have to do functional assays to prove that the change is actually causing the disease. So as you can imagine, we have sequenced 500 genes for 600 different types of diseases or types of the rare diseases. And you can imagine the incidence of rare diseases is slightly higher in India or in the Middle Eastern countries, mostly because many groups in India really have close marriages, like first cousin, second cousin. And when that happens, the disease-causing genes actually accumulate. And there are many people, like, some certain communities in Bombay, there are very high incidence of actually rare diseases. So it is very easy to find rare disease when you are really studying consanguineous marriages. But what we are finding now, after sequencing almost 40, 50,000 samples, we are finding that it's getting harder and harder to find the cause of the disease. And that's because I think we don't find that disease could be somewhere else, or could be protein change, could be glycosylation change. I don't want to be too technical. There is also methylation changes. So we are exploring all those options. So National Institute of Health, actually, so one of the reasons I told you the technology is changing. Currently, it's costing like $1,500 to do the whole genome. So this is a technology which is really upcoming called Oxford Nanopore. This technology pretty soon, I'm pretty sure in next 3 to 4 years, will bring the cost down to like a couple of $100 or $300. So when that happens, you can trust me, every baby which is born, at least in developed countries, will have their genome sequenced. And whether baby wants it or not, there will be list of the diseases that baby might have during the lifespan. So the technological advances are actually, so in US, I think they are spending like tens of millions of dollars to do just the research on rare diseases. And I'm lucky enough to get one of the grant. It's a $12 million. And so we have been, through this grant program for last 6 years, we have been sequencing many rare diseases. And so the whole idea here is that even if you take a cystic fibrosis, which is one disease, there is a lot of genetic heterogeneity. That means there are many genes which contribute to the same disease. So then it becomes like I mentioned here, when you sequence more and more and more patients, you find all the genes which are associated with a particular disease. So it's very important that we continue sequencing, continue identifying these rare disease patients, take their blood, isolate the DNA, and sequence them so we can completely identify all the genes which cause for a particular disease. And this is why the National Institute of Health has decided to do the worldwide collaboration. So we are gathering samples all over the world, and I'm definitely making an effort to get samples from India. And so there is a precision medicine effort now. What is precision medicine? You must have heard that there are many drugs which don't work on all patients. This is a very common thing, So, or some people have more side effects than other people. So this is why, because we all are different. Genetically, even though we are 99.9% identical, there is a 0.1% differences, which makes who we are, how we look, how we think, what's our intelligence, what's our thing. So these 0.1% differences are responsible for how drug interacts with our body. So the new efforts will be made in such a way that drug will be given based on everybody's differences. So we'll be sequencing the genome, we'll identify which gene is causing the disease, and the gene will be treated rather than the disease. Because there are many cases we have found out that melanoma, for example, not all melanoma patients really respond to one treatment, and that's mostly because there are different genes which are causing melanoma. So if we identify the different genes, then we can also give a different treatment to every patient. And that's what is called precision medicine. And President Obama announced it, and he started with my lab at Yale that we started the precision medicine efforts. And I think we are mostly doing it cancer, but we're also making an effort to do it for the rare diseases. So this is a very common thing in US doing the genetic testings. I don't know whether you have heard of 23andMe. This was in cover of Time magazine. You can order any test you want. So I will skip that. So I want to talk about what is the future with the technology. As I told you, US government is spending hundreds of millions of dollars every year to sequence the human genome and trying to identify the cause of the disease. As you know that the first step when the car breaks down is to first find out what is the cause, and then you can decide how to fix it. The same thing is with our diseases. First, we need to find out what is the cause of the disease, and then we can design the treatment. There are many diseases for which when we sequenced it and find the cause, we realize, oh my God, the treatment already exists. But we didn't know that this treatment will be applicable for a particular disease because we didn't know how the disease is happening. So newborn screening is a public program in all developed countries, and there are many states in India also have that program. So every child born within a day or two, it's screened for 50 known disorders. This is called So right now, the type of technology which is used can allow us to screen only for 50 diseases. But the technology which I'm talking, sequencing the genome, we can actually test for all 3,000 genes. And we can actually take the blood from a 3 months pregnant mother, not the amniotic fluid. Take the blood from the mother, take the blood from the father, and determine the baby genome. And we'll have list up all the defective genes way before the baby is born. So we can actually determine the plan of action when the baby is born, in case the baby has the disease. So I just want to tell you one thing, this newborn screening program is one of the most successful program, has saved like 60,000 babies. This slide is a little bit old, but it's amazing how this program works. And I also want to tell you what future is there. Newborn screening program is already being actually implemented in United States, but there is something which is very exciting for, especially for the rare disease community. So for example, even if you find out one base pair which is changing, which is causing the disease, so what? Because you really can't change that base pair in all the cells in our body right now. But the CRISPR is the technology which will allow us to make that change. It will allow us to cut the part which is really gone bad and replace it with the good part and stitch it together. However, this has to be done probably in vitro fertilization. You can do that only when there is egg and sperm, or only when the egg and sperm unite and form one cell from which entire human body is developed. At that stage, you can make that change. You can't do that more. So what I'm saying is all the genes we are identifying which cause rare diseases may not help current population of the rare disease now, but it will help their children. And with that, I want to thank you.
Thank you. Thank you. That was an extraordinary, overview of the blue sky work that countries like the US can afford, and I think Yale is obviously a great center for that. We can again pick up with more questions later. Meanwhile, let me request Dr. Navneetham to bring us down to India and also into the rural part of India, because I think his work with rural India and rare diseases in rural India is actually quite exemplary.
Where is the gentleman?
Nations